Hello everyone, this is Water and Soil Microbiology Practical Lecture 4, Jihan University De Hoek Medical Microbiology Department. So today we're going to do an experiment about the benefit bacteria in the soil. So the soil is the richest environment with bacteria and microorganisms in general, of course. So, and these bacteria, most of them have an important role, and the importance in the all of the biochemistry and very benefit for plant, for animal as well. So do a lot of biochemical reactions inside the soil and among microorganisms in the soil. The first and the important one of the benefit of microorganisms is decomposition. Decomposition is the complex of organic material to the simplest material. Some of the bacteria do the fixation of the nitrogen. If you remember in the theory, we have the biogeochemical cycle, and the main of the biogeochemical cycles was the nitrogen cycle and carbon cycle. So related to the nitrogen cycle, today in our experimental, I mean the practical. So the fixation of the nitrogen. So what are those bacteria? What they do? They, for example, the oxidation of ammonia occurs in two steps. First, ammonia is oxidized to nitrate, nitrosofication. The most microorganisms do it, it's nitrosomonas and nitrosococcus. The second step is these NO2, the nitrate, oxidized to nitrate, from ammonia to nitrate, from nitrate to nitrate. Okay, so nitrobacter, like the bacteria which do this. This process, some of the bacteria is make a relationship with the plant, with the root of the plant, and do the fixation of nitrogen from the air to get benefit, and to give this to the plant. So this is one of the process in biological nitrogen cycle and fixation, which takes place in the noodles located on some of roots of some plants, like the family Fabaceae, which is in Arabic بيسموه الفصيلة الفولية. Okay, in Arabic بالكردي بجنب بقلايا وصى يعني. Okay, nodules formed in the result of infections. It is like an infection of the root by the soil bacteria. The main bacteria of which do this and related to this relationship, it is Rhizobium. So this is why which in today is our practical on Rhizobium. So the bacteria here, look, as you have seen this here. In noodles here, it's like red or between red and brown noodles located on the root of the plant, and they do, of course, the fixation and the nitrogen. What are the physical and behavior features of the genus Azotobacter? This is another, another bacteria which do this. These bacteria live free in the soil, and some of them located on the root of the plant, of course. Require oxygen to grow, of course, live in the soil. Rod-shaped and negative, of course, it's gram negative. May it's capable to movement by flagellum, very large in size of bacteria, and can produce some pigment. So these pigment, as you have seen in the media, if you culture these on the media like general media and others media, of course selective media for them, you will see the mucoid convex large colonies and the colored colonies, like it's white, brownish, as you have seen in the pictures, white, brownish color, or sometimes colorless. Black sometimes, yeah, it is. This is the pigmentation process. I mean, under microscope you will have like this. Of course, this is Azotobacter, and our practical today is one of the bacteria, which is Rhizobium. I'm going to show later in this practical, of course, and you will do and work in hand in the lab. These Azotobacter, as you have, they do any symbiotic. I mean, it is symbiotic nitrogen fixers. As I have explained now before. The symbiotic is a relationship between the plant and the soil and the microorganisms, and this is do this fixation of nitrogen for soil and for plant as well. The main symbiotic, as I said, it is Rhizobium. Rhizobium is a gram-negative rods, motile, and by flagella, and then look, this is this root. Have this nodulus, and this nodulus is very full with Rhizobium and other bacteria like Azotobacter, of course, but especially Rhizobium. The benefit of Rhizobium, they improve soil health, provide stress tolerance in the soil and the plant, reduce dependence on fertilizer, and fertility restorer. So improve the nutrient availability for plant and soil, of course, and biological it's nitrogen fixation, of course. How can I do the isolation of the Azotobacter? So this species isolated from the soil by do this dilution method technique. It's a dilution plate technique on the nitrogen-free medium. This is the selective medium for Azotobacter. It is Jensen's media and Ashby mannitol agar media. This is the selective. These are the selective media. So after 48 hours or 3 days, maybe. 3 to 4 days, and if it incubated in the 30 centigrade, so flat, soft, milky, and mucoid colonies will grow on the surface of the plant, as you see. It can sometimes isolated in the lump of soil on nitrogen-free nutrient medium, of course. So this is the Azotobacter. Related to our practice today, it is Rhizobium. So the student, you will bring the root and the plant with the nodulus, and this nodulus we will gonna cut the roots of any legumes of the plant and wash it by tap water. Select and cut visible large pink nodulus. You're gonna to select the pink nodulus. We're going to transfer this to clean glass slide and crush it carefully. We can use the cover slide, pressing it on the slide, and then when you see the milky suspension get out, or check it, so they then mix with water drop. You're going to do mix and do the gram staining. So it's a gram negative, of course. Under microscope, after staining by the oil glasses, oil lens, oil immersion, you will see the Rhizobium like Latin letters like Y, X, Z, and T. So as you have seen in this picture. So this is our practical, today, and you will work in hand in a group, of course, and everything is on the bench in front of you. You will do it in by the group and work in team to do this, and of course you will bring and write the report about this practical later. Thank you for your listening.